Hi, I'm Dawn Eaton, the CEO of Susan G. Komen Central Tennessee. And today I have with us Dr. Ben Park. So welcome, Dr. Park, and thank you for spending time with us. We want to spend just a few minutes today and introduce our audience to the research that Susan G. Komen is doing broadly, and then very specifically, what's going on right here in our own backyard at Vanderbilt-Ingram Cancer Center.
Well, thanks for this opportunity, Dawn. It's really a pleasure and an honor to be here and talk about Susan G. Komen and the great work that's being done, specifically in the research realm. So I'm a Komen Scholar, and what that affords us to do is really work with the chief scientific officers and advisors to think about the directions that Komen really wants to go forward in terms of breast cancer research. And that's both at the clinical level, the laboratory level, and everywhere in between. And so that's the really both just amazing but also humbling part about our jobs, is that we get to really set the direction in terms of what we feel are the unmet needs and what would be the most immediate deliverables. We have a bold goal of trying to reduce morbidity, mortality, and I think that that's the way our research really should be driven. Specifically here in Nashville, where I'm working at the Vanderbilt-Ingram Cancer Center, there are a few of us who are Komen Scholars, and that also affords us the opportunity to apply for research grants. And my grant's really looking at how certain breast cancers may carry certain DNA mistakes or alterations that might make them susceptible to these immunotherapies. And I think a lot of people know about these immunotherapies. For example, Jimmy Carter, our past president with metastatic melanoma, has really benefited from these new therapies and is alive and well and possibly even cured. We don't have the same successes with those same types of immunotherapies for breast cancer yet. And so what we're really trying to do in my lab and my research program is look at certain types of breast cancers with certain types of DNA alterations, or what we call mutations, that might further predispose those particular breast cancers to immunotherapies.
That's pretty exciting. Where are you in your research? Like, how far along are you on this particular project?
So for me, this is a really great opportunity as a proof of principle to take stuff from the lab and move it into the clinic. And so one of the things we did, the genes and the alterations that we're looking at seem to create a whole different, what we call transcriptome. You don't have to know what that means, but it changes all of the different DNAs. So even though it's one DNA mistake, it actually changes everything that all the different sister DNAs, if you will, make in terms of their products. That's really what has led to immunotherapies working. This is work done actually by my former boss at Johns Hopkins, Bert Vogelstein, that really showed the more protein changes you have in a cancer cell, the more likelihood that you're actually going to get a response from these immunotherapies. And so we kind of stumbled in on this and discovered that, hey, there are some breast cancers, not many, maybe about 3 to 5%, that have these particular DNA alterations that seem to change all the proteins in the cancer cell. And we've hypothesized, based upon retroactive data in other cancer types, that this would lead to a better response. What we've done now, though, is to take this to the clinic. We are doing what we call a virtual trial. And we haven't actually opened this yet, but we're about to. And so colleagues at Hopkins took over this from me when I was there as a Komen Scholar, and we're going to partner with them and bring that trial here. And what we're going to try to do is reach out to patients who have this DNA alteration. So nowadays, a lot of times in metastatic patients, we get a biopsy and we send it off for what we call next-gen sequencing to look for the DNA alterations. And we've already seen a couple here in my short time at Vanderbilt where patients would have these mutations. So you could never do a real trial if you only have 2 or 3 of those mutations at an institution in a year. But what we're hoping to do is, by collecting everyone's data in a large database, kind of advertising this on social media. This is really going to be one of the first trials to do this. It's merging a concept that some people have done for databases, as well as something that we call like match trials, like ASCO's running. Putting those two concepts together to try to capture as many patients as we can, have them upload their reports into a secure database, and we're going to go through them, and if they qualify, we're going to contact them and say, hey, you could potentially get this drug, because this drug's already FDA approved for other cancer types. And so
that's exciting.
So this is something that we're really hoping will be a new way of thinking in terms of how we can get drugs approved with a trial for other types of cancers. So you have this one drug that's already approved for a certain type of cancer. This would allow us hopefully to pivot and say, you know what, for these rare cancer or for these other cancers that rarely have this particular DNA alteration, we might be able to get an FDA approval now if this trial is successful.
Now, is that part of Komen's big data project, or is that separate from the project that Komen's working on?
It is separate, but I hope to actually at some level combine them as well as other national databases, because I think sharing of data, I think most of us would agree who are in that business, is a good thing. And it just benefits everyone from, you know, researchers to physicians and particularly our patients. So I think it's on us and behooves everyone to really make sure data gets out there, because it's such a valuable commodity that should not be siloed and needs to be shared.
Well, and that's something that perhaps we can talk about even on another interview, is we want to introduce our audience to what all Komen's doing. And yep, we're just talking about one piece of it today.
Right, that's
and,
right.
When you were talking about Komen Scholars a while ago, I think it's important for people to realize we've got 51 Komen Scholars across the country.
Yeah.
There is a lot of work being done, and I believe that every major breakthrough that's ever come through in the United States in particular, there's been Komen dollars behind that. So yep, I think the research is so important, and it's really exciting to realize that it is right here. So much of it is right here in our backyard. I thought our audience would be interested in knowing that Komens Research investment has been more than $1 billion in more than 2,700 research grants and more than 500 clinical trials since our inception in 1982.
That's amazing. That is really amazing. And kudos to Komen. You're absolutely right. The work that Komen has done, though, you know, beyond the research, it's been incredible, you know, making access better and easier. But research is really what drives the cure. And I think most people recognize that. And we are committed to curing this disease. And I think that for me has always been front of mind, and why, you know, I really want to help push this forward.
So when you were talking a minute ago about this being kind of a virtual clinical trial, could you speak just a moment to clinical trials in general and why they are important for breast cancer patients?
Absolutely. We can't really know if a drug works well on a kind of broader basis unless we do a clinical trial. It has to be really well controlled, meaning that there has to be a placebo group. So we do trials in different phases where we test for things like side effects, look for hints or signals that it could actually work. But to get it really to that next level where it's approved and everyone can get it really requires a prospective randomized trial, because the statistics aren't valid otherwise. And it's kind of sobering to me that we keep learning this lesson over and over again. By that I mean there was recent data that just came out at one of our big conferences that this drug Ibrance, which has gone through the ringer for metastatic breast cancer patients, we know it works really well, and we all thought and presumed, now that we're going to move it to the early breast cancer space, looking to see if it can actually cure more patients, that it was going to work because it has such a powerful effect in the metastatic setting. But the early results that were just released suggest that, in fact, it's not going to be any better. And that's really sobering and disappointing, obviously. And who knows, maybe with longer term follow-up, we'll see a signal. But I think for right now, many of us were surprised, including myself. And it's a lesson that we keep learning. This is why we do trials. This is why we have to have rigorous controls and make sure that at the end of the day, given the cost of new drugs, given the toxicities that some patients will experience, it does us no good as a field to prescribe something that's not going to work.
So, Dr. Park, I think that the term clinical trials, you know, people probably had heard of clinical trials before, but I'm finding with COVID-19, with talking about the vaccine and all of that, that people are becoming more familiar, you know, with the term, and the fact that it's really not a short-term process. You know, the number of layers to clinical trials. Is that correct?
It is absolutely correct. And it's very complicated in oncology or cancer clinical trials research, because we have to make sure that we're not ever denying standard of care. And so we have, certain drugs that are standard of care. We can't deny the standard of care to the control arm. So our trials are always designed that we're stacking more drugs onto the standard of care versus placebo. And I think that that's makes it even more and more complex as we gain more traction into trying to cure this disease with better drugs. And so we've even now started to look at de-escalation trials, meaning that if there's an early stage breast cancer or the features look like it's going to do fine without so much therapy, can we now safely design trials that say, hey, what if you have a standard of care and you have a little bit less than a standard of care, because the data, if we examine it retrospectively, would suggest that this subgroup doesn't really get much benefit from like 3 or 4 drugs as opposed to 1 or 2. And so that's a big concern of us, because we're just piling on toxicity the more drugs we add. And so I think our field is really evolving in how we're thinking about treating patients more precisely. The COVID has been great in the sense that, it's a new virus, and so there's no real standard of care. And so you can get the trials done quicker and better and faster in that regard. Now, it's again a lesson learned. There's all this hype about hydroxychloroquine being some sort of preventive or treatment effect for the COVID. But we as a country and many institutions, including ours, did rapid clinical trials looking at that question. And you may have heard that, in fact, the results were negative. And if patients took 2 drugs, hydroxychloroquine with something that we call azithromycin, which is a common antibiotic, they might have even done worse. And it looks like they actually did do worse. So again, this is why we do clinical trials. We have to do it in the context of a control arm to know that, are we doing actually anything better? Is it the same, or is it possibly even worse?
What about, speaking of COVID and clinical trials, I believe you all have been doing some research, haven't you, around COVID and breast cancer patients?
Yeah.
Can you just give us an update?
Yep. So ours were, at least at Vanderbilt, were more geared towards not just breast cancer, but cancer in general, and patients who were tested positive for COVID. And so my colleagues, Dr. Jeremy Warner and Brian Rinney, they really led the effort here at Vanderbilt, particularly Dr. Warner. He's got skills in bioinformatics, and he actually was the first to really say, we should be collecting this data, talking about big data and sciences that we were mentioning earlier, and that's kind of what he does. And so he created very quickly a secure database to start capturing our data and expanded it beyond Vanderbilt. In fact, so it's called CCC19. The results were just published in The Lancet, which is one of our premier medical journals. But I like to say, tongue-in-cheek, that it went viral. And it just incredibly quickly went across the nation and the world, where all these institutions and hospitals joined so that we could collectively look at our data. And, we found that cancer in general is probably a state where you're more susceptible to the bad effects, or not that they're good ones, but the effects of COVID virus in terms of dying from the disease. So there's a lot that we still don't know. There's a lot we're learning now as we go forward, but having a way to centralize this data and follow it prospectively is a huge, huge win for us as oncologists.
Well,
and congratulations, obviously. Oh, thanks.
Congratulations on your work. Listen, I just want to thank you so much, both for your support of Komen, for the research that you're doing, and I think just for your heart for this work. You put a lot of passion into it, and I just want you to know how much we appreciate it. As we wind up today, I wanted to share with our audience, as I mentioned in kind of the opening comments, that so much research is actually going on right here in Middle Tennessee. And I wanted to share that since the year 2000, researchers at Vanderbilt have received $14.5 million. That was for 37 grants, and that was from Susan Komen. And currently we have 8 scientists right now working on about $4.9 million in active research grants. So thank you, and we wish you the best of luck.
Oh, thank you, Dawn. It's always been my pleasure to help when I can. And you're right, I'm so passionate about Komen and finding the cure. So thank you again.